Hello, and welcome to my laboratory. Today, I will be teaching you about glycolysis in alcohol and lactate fermentation. In glycolysis, there are two steps in glycolysis. The first is the energy investment step, in which two ATP molecules are necessary to get started, which ATP stands for adenosine triphosphate. And the two ATP combine with the 6-carbon glucose molecule to make this 6-carbon molecule with 2 phosphates on each side, and then that molecule then splits and forms 2 3-carbon molecules, and these are called G3P, and that is the end of the energy investment step, which that brings us into the energy harvesting step, in which NAD plus is changed into NADH with each G3P molecule, and then a phosphate is added, which creates this 3-carbon group with 2 phosphates, and that is known as BPG. And this process here is when this is called oxidation of the G3P molecule, and the NAD plus received a high energy electron, which is what turned it into NADH. And then after we have the BPG, a phosphate group is added with the ADP, which creates ATP, and that's taken away from the BPG, which creates this 3PG, which is the 3-carbon molecule with a phosphate as well. And then when this ATP is created, that is known as substrate level ATP synthesis. And the next step is when H2O is lost from the G3P molecule, and which leaves us with this other 3-carbon molecule with a phosphate known as PEP, and that is known as oxidation once again. And then ADP is once again changed into ATP, so we gain another 2 ATP here, and that results in just this 3-carbon pyruvate because we lost the phosphate, which created the ATP. And that is the end of glycolysis, which we lost 2 ATP to begin with, and then we gained 2 ATP here, and then we gained another 2 ATP here, which results in a net gain of 2 ATP with a total of 4 produced and 2 lost. Or, and then next, if there's no oxygen present, this will result in alcohol or lactate fermentation, depending upon if it's in an animal or a plant. And this, from here to here, this is the same process that is here, just shown once again for fermentation. And then, so after the glycolysis, we end with the 2 3-carbon pyruvate molecules, which then, if it's an animal, it'll go this route and be changed into 2 lactate molecules, or if it's a plant, it'll release 2 CO2 molecules and 2 ethanol alcohol molecules for if it's a plant. And the 2 NADH that are created are just recycled back into NAD to be used once again. And it's the same as glycolysis, so 2 ATP are used to start in the energy harvesting, and then 4 ATP are created through glycolysis. So it's, again, same as glycolysis, a net gain of 2 ATP molecules. And that concludes today's lesson.